Hello everyone, welcome back to my channel Excellent Protein X. In this tutorial video, I'm going to talk about the granzyme and perforin mediated apoptosis by cytotoxic T cell. Let's begin. So this is the interaction between T cell and target cell. So as you know that the T cell having the T cell receptor. So the T cell receptor, it recognizes antigen presenting by the major histocompatibility complex of target cell or antigen presenting cell. And then T cell get activated and it release different cytokines and perforin and granzyme. So perforin is actually the pore forming protein and the granzyme is related to the serine and protease family. So upon secretion of this perforin and granzyme, it get into the target cell and then granzyme it activate the apoptotic pathway. Then target cell it eventually die undergoing apoptosis process. So there are different varieties of the granzyme. In humans, there are granzyme A, B, H, K, and M, and in mice there are a lot more varieties compared to the humans. But granzyme A and granzyme B, these are mostly common and abundant. So the different varieties of this granzyme and perforin, these are packed together into the cytotoxic granules of cytotoxic T cell. So next I'm gonna discuss about the activation of the granzyme mediated apoptotic pathway into the target cell in details mechanism. So the cytotoxic granules contain the different varieties of the granzyme and perforin, and when this cytotoxic T cell it get activated, then these granules it release. And then perforin it create pore into the cell membrane of the target cell. Then the granzyme it pass through this pore and get into the target cell, and then the granzyme it activates the apoptotic pathway. So the granzyme B it directly activates pro-apoptotic BH3 member of the BCL2 family, that is the BEAD. So BEAD is actually BH3 interacting domain death agonist. So BEAD it work together with pro-apoptotic BCL2 family proteins. Those are BAX and BAK. So as a result, the leakage of the pro-apoptotic mitochondrial mediators, that is the cytochrome C, into the cytosol, and then cytochrome C it release and activates procaspase 9, and by binding to apoptotic protease activating factor 1, that is the APAF1, then procaspase 9 becomes mature caspase 9, which contributes to form apoptosome, and it activates the downstream caspase 3. So the caspase 3 to activated caspase 3, these are activated by the apoptosome. And then activated caspase 3, it's able to cleave the specific substrate like ICAD. So ICAD is the inhibitor of the caspase activated DNase. So this is the inhibitor. So the ICAD and this caspase activated DNase associated together, but the activated caspase 3 it cleave the CAD, and then CAD it translocate to the nucleus, and then it fragment the DNA of the nucleus. So beside BEAD, granzyme B it can inactivate MCL1. So MCL1 protein, this is the member of the anti-apoptotic BCL2 family, and this MCL1 it can release the pro anti-apoptotic protein, the BEAM. So upon inhibition of this MCL1 by the granzyme B, the MCL1 cannot activate BEAM, and then this also the granzyme B it can also activate caspase 3, So this is the another mechanism that granzyme B, it can mediate caspase-3, as well as it also activate the procaspase 8 and eventually the caspase 8. So caspase 8 is activated by the granzyme B. This is an indirect pathways. And also the granzyme M, it also activate granzyme B by inhibiting PI. So PI is actually the proteinase inhibitor 9 that actually inhibit granzyme 9. So granzyme 9, it actually inhibits PI, then the PI's action is inhibited, and indirectly granzyme M, it activates granzyme B's function. So, and also the granzyme M, it also works on ICAD, and also granzyme M also it cleave CAD as like granzyme B. So this is all about the granzyme B and granzyme M, their contribution onto the apoptotic pathway activation, and also the granzyme A, it has the major role in contribution for the apoptotic pathway activation, but its mechanism is little different. So granzyme A actually it induces loss of mitochondrial inner membrane potential and release ROS. So the reactive oxygen species or ROS, it generates single-stranded DNA mix into the target DNA, and in response to the ROS, the ER associated set complex, those set complex contain some group of proteins, that is HMG2, PP3, AP1, TREX1 set. So this kind of group of proteins, those are known as set complex. So in response to the ROS, this set complex, it translocate from the endoplasmic reticulum to the nucleus. But out of these set complex proteins, these are the set complex proteins. There are a group of proteins, but out of those proteins, there are 3 different proteins. These proteins has major role in DNA repairing. So the granzyme A, it actually inhibits those proteins functions. So these 3 proteins actions inhibited by the granzyme A, then eventually the DNA repairing inhibited into the target cell. So this is how granzyme A, it has the contribution on the apoptotic pathway activation. So this is all about the granzyme mediated apoptotic pathway activation. So I hope this video will be helpful. If you like this video, kindly hit the like button, share it, and if you really like, or if you really think that this video is helpful, then please subscribe my channel. That encourages me to make more videos. Thanks.